Hello, I'm Ash Dugar, the Global Head of Medical Affairs at Dyne Therapeutics. First, I would like to thank Tally and the entire Little Steps organization for inviting me to this chat to update the Duchenne community on the progress of our Duchenne program, and specifically Dyne 251, our clinical candidate designed to treat patients with Duchenne who are amenable to exon 51 skipping. As always, it is a true privilege to speak with the Duchenne community. These are our forward-looking statements. Please refer to our website if interested in more details. At Dyne, it all starts with our mission. We're laser-focused on delivering life-transforming therapies to patients with serious muscle diseases, with Duchenne being a priority. Our mission is core to who we are and what we do every single day. We're incredibly excited to inform you that we have started dosing participants in our Dyne 251 program, and our focus today is on sharing an update on our clinical trial progress and details about our clinical trial design with Dyne 251. Over a short few years, we have built a robust portfolio focused on rare muscle diseases. Myotonic dystrophy type 1, or DM1, and FSHD represent opportunities to develop first-in-class therapies, and in DMD, we are building a global franchise starting with our exon 51 program. And we will follow this with programs for exons 53 and 45. This slide provides an overview of Duchenne, which you all know very well. Our goal is to develop a best-in-class exon-skipping treatment targeted to skeletal, respiratory, and cardiac muscle that greatly increases dystrophin expression and has much less frequent dosing than current agents, with the hope of potentially stopping or reversing disease progression. Here, I will take a couple of minutes to describe the FORCE platform. The FORCE platform drives our efforts to develop and deliver muscle-targeted therapeutics. It is expressly designed to address one major issue in muscle diseases, which is efficiently and reliably delivering antisense oligonucleotides that target the genetic basis of diseases to skeletal, cardiac, and smooth muscle. As you can see, Dyne's FORCE platform, or our scientific platform, consists of three parts: a proprietary antigen binding fragment, or an antibody fragment, or what we call a Fab, to allow targeted delivery of the antisense oligo, or ASO, to muscle, a clinically validated linker, and an antisense oligonucleotide, or ASO, selected to target the genetic basis of disease. On the left part of the slide, you can see a Y-shaped structure, which is a monoclonal antibody. Well, we chose the top portion of this antibody, our Fab, as I mentioned, because we believe it offers powerful advantages over using a monoclonal antibody, including its smaller size, lower overall protein load, and the potential for enhanced tolerability. The goal is ultimately to enable targeted, effective, and efficient delivery of therapeutic molecules to skeletal, respiratory, cardiac, and smooth muscle. On the bottom part of the slide, you can see the linker. We selected the Val-Cit linker for our platform based on its stability and clinically validated safety and efficacy in approved products. We believe this further de-risks our FORCE platform. Now, we utilize the same Fab and linker across all of our neuromuscular programs, including Duchenne. The versatility and modularity of our scientific platform provides flexibility to attach different types of antisense oligonucleotides that are rationally selected to match the target biology of each disease. Where dystrophin restoration is the key, as is the case in Duchenne, it's critical that we select an exon-skipping phosphorodiamidate morpholino oligomer, or PMO, that targets the specific mutation in the DMD gene. So in our current study, we are using a PMO that allows for skipping exon 51. The mechanism of FORCE is different from other approaches. We are essentially using the cell's natural biology to deliver our therapeutic to muscle cells. Our Fab binds tightly to something called the transferrin receptor 1, or TFR1. TFR1 is highly expressed across the surface of muscle cells, including skeletal, respiratory, cardiac, and smooth muscles. Now, the Fab acts as a kind of key that unlocks the TFR receptor and allows our PMO to enter muscle cells and target the nucleus of the cell so that we can address the genetic basis of Duchenne. In short, we are able to harness what already exists on the surface of muscle cells to deliver our therapeutic to muscle with this receptor-mediated mechanism. By targeting muscle specifically by using TFR1, we have the potential for a wide therapeutic index, hopefully resulting in less frequent dosing and chronic administration. On this slide, we show our force platform in the MDX mouse model, a standard animal model in Duchenne. These MDX mice have a mutation in exon 23. You can see that force resulted in robust dystrophin production across key muscles affected by Duchenne after just a single dose of 30 milligrams per kilogram, and then the mice were evaluated 4 weeks later. The top half of this slide shows some of our data demonstrating robust levels of dystrophin expression across these muscles. We're able to achieve high levels of dystrophin expression, approximately 50% in the quadriceps, 90% in the diaphragm, and nearly 80% in the heart. We were extremely excited to see these levels of dystrophin restoration, especially in the heart and diaphragm, where current PMOs have had significant limitations. On the bottom half of the slide, we show dystrophin localization in the sarcolemma muscle membrane after just a single dose. This localization is important to see to provide confidence in potentially seeing functional benefit. And we saw profound results, including about 70% dystrophin-positive fibers in the quadriceps and about 80% in the diaphragm and heart at 4 weeks. Importantly, our force platform has shown to have durability of effect. This slide shows our data on dystrophin-positive fibers in the quadriceps out to 12 weeks. So even out to 8 to 12 weeks, we saw sustained localization of dystrophin to the sarcolemma and the quads, and we've also seen this in other muscles after just a single dose. Next, we evaluated Dyne 251, our clinical candidate, in non-human primates, or NHPs, or in monkeys. These studies are a key step in generating preclinical data before going into clinical trials. We measured exon skipping in the quadriceps, heart, and diaphragm. Please note that in monkeys, we cannot look at dystrophin restoration because they are healthy animals. The key message is that with our force platform, time on therapy matters. Over a 2-month period, we observed robust exon 51 skipping across muscles. After 5 weekly 30 milligram per kilogram doses, Dyne 251 demonstrated 52% exon skipping in the diaphragm, 43% skipping in the heart, and 18% skipping in the quadriceps. Again, we're really excited to see this kind of exon skipping in the heart and the diaphragm. We also evaluated Dyne 251 in 5 and 13-week toxicology studies in monkeys. We saw that Dyne 251 had a favorable safety profile with no dose-limiting toxicities observed after weekly doses up to a maximally feasible dose. In short, we're very pleased with the safety and tolerability profile as we advance to the clinic. This slide summarizes an extensive amount of work that we've done in animals and in patient cells to be able to move forward into the clinic. And currently, our DELIVER global clinical trial is recruiting. Prior to starting our Dyne 251 trial program, however, we proactively obtained expert feedback. We engaged key opinion leaders or KOLs from various disciplines, including pediatric and adult neurology, cardiology, physical medicine rehabilitation, pulmonology, and physical therapy. Importantly, we have gathered this feedback from KOLs across the globe. And these KOLs, both in the US and outside of the US, have great experience running early and late stage clinical trials, and they provided advice on our overall clinical development program, including our clinical trial design, the patient population, biomarker and functional endpoints, and key safety considerations. And they've also helped us to extract learnings from natural history studies. It is clear that KOLs believe dystrophin continues to be a key endpoint for this condition. We've also received advice directly from patients and caregivers, as well as global advocacy leaders, such as those from Israel, as part of the Dushen Community Advisory Board, comprising of advocacy leaders from several countries around the world. They provided insights based on their living with and being the voice of the Duchenne community, as well as experience with past and current trials. And this input has been critical in ensuring a patient-centric design and has included, but is not limited to, key considerations for choosing a clinical trial, review of and input to our informed consent form, feedback on biopsies and functional outcomes, identification of the most burdensome or difficult parts of the trial, and how to ensure patients and families are supported throughout the process. And based on this extensive feedback, we have developed the proposed clinical study for Dyne 251 in patients amenable to exon 51 skipping. And I will describe the key elements here. Our trial for Dyne 251 is a global multiple ascending dose, or MAD, placebo-controlled study with a long-term extension. It's important to note that following the placebo-controlled portion of the study, all patients will be able to receive active treatment. We are enrolling patients with Duchenne who have mutations amenable to exon 51 skipping therapy. Our goal is to enroll approximately 46 participants ages 4 to 16 years who are either ambulant or non-ambulant. We plan to evaluate the following outcomes in the DELIVER trial: safety and tolerability, dystrophin by Western blot, muscle function that's relevant to both ambulatory and non-ambulatory participants. So that would include assessments to help us understand changes in lower and upper limb function, as well as lung and cardiac function. And importantly, we will be dosing monthly or less frequently in the clinic. With the help of our expert advisors, we are very confident that our study design is robust, will measure key outcomes, and is striving to manage the burden on families. And finally, we have tried to take every opportunity to listen to the Duchenne community on their needs for next generation treatments. We are working urgently to deliver for the community, including families and healthcare providers, to bring a potentially transformative treatment forward. Thank you to the families for being generous with your thoughts, advice, and for the sacrifices you continue to make. On behalf of Dyne, I would like to thank you all for your time and attention in listening to this presentation.